Now we will study something in detail about restriction endonucleases. Restriction endonucleases belongs to the class of endonucleases. Endonucleases are those enzymes which cleaves your DNA molecules. That means they put internal cuts. Now depending on whether they cleave only one strand of DNA or two strands of DNA, there are different types of endonucleases. In our DNA technology, we are basically interested in restriction endonucleases. Okay. Now we have studied what are endonucleases. They put cuts in your DNA strands. Now why the term restriction endonucleases? Because they have the ability to identify specific your set of nucleotides. When your restriction endonucleases will come across that specific set of nucleotides, then and only then they will cut your DNA strands. Okay. So this is how your restriction endonucleases work. They basically cut your covalent bonds between your two strands of DNA. Okay. So you have two strands of DNA here. When your restriction endonucleases, for example, EcoR1 is an example of restriction endonucleases. So when your EcoR1 comes across this particular pattern of nucleotide, it will put a cut and it will cut your G between your G and your A. It will cut here. Okay. So because of that, now two strands of DNA has become separate. Okay. Now there are three types of restriction endonucleases: type 1, type 2 and type 3 restriction endonucleases. Mostly we go for type 2 restriction endonuclease. Okay. Restriction endonucleases usually recognize a sequence which is called as a palindrome sequence. Okay. Now just have a look here. This is a palindrome sequence. Now what is a palindrome sequence? If you read it in the forward direction or if you read it in the backward direction, the arrangement of nucleotides is the same. So that is called as your palindrome sequence. So your restriction endonucleases will always cut when it comes across this palindrome sequence. Okay. Now this palindrome sequence is either 4 base pair long, 6 base pair long or 8 base pair long. Okay. Now your restriction endonucleases can either cut and make a blunt end or it can make a sticky end. So what is that? So these are examples of some restriction endonucleases and the types of cuts where they are making. So you can see that either they make it at one end or they make it exactly at the center of that sequence. Okay. So this is the nomenclature by which you give naming to your restriction endonucleases. So example here is EcoR1. So it indicates that that restriction endonucleases is obtained from your E. coli, from the R strain, and it was the first that was identified. So this is the nomenclature for restriction endonucleases. Okay. Next is the types of cuts that the restriction endonucleases make. So you can see two examples here. First is your EcoR1 and second is your TVU2. So you can see that restriction endonucleases, when it comes across this sequence GAA-TTC, it will cut your G and A. Okay, and you will get something like this. Okay, so you can see that your restriction endonuclease has cut like this. So this is called as a sticky end. You have something to stick to this end. So that is why it is called as your sticky end. Okay. if you see this example, that is PVU2 restriction endonucleases, when it comes across this sequence, that is CAG, CTG, it will make a cut exactly at the center, dividing these two strands into half. Okay, so you can see that there is no sticky end here, nothing is protruding out, so that is called as your blunt ends. Okay, usually we prefer endonucleases which produces sticky ends because it is easier for the ligase to seal the sticky ends rather than the blunt ends. Okay, if we somehow need to use a particular restriction endonucleases which produces blunt ends, in that case these blunt ends needs to be converted into sticky ends by using certain linkers or adapters. Okay, so these are some examples of restriction endonucleases and the types of cleavage that they